Researchers discovered that a certain molecule in broccoli interacts with a receptor in mice to promote gut health. Broccoli is known to be beneficial to our health. For example, research has shown that increased consumption of the cruciferous vegetable decreases incidences of cancer and type 2 diabetes. In a recent study, researchers at Penn State found that broccoli contains certain molecules that bind to a receptor within mice and help to protect the lining of the small intestine, thereby inhibiting the development of disease. The findings lend support to the idea that broccoli truly is a superfood. The wall of the small intestine allows beneficial water and nutrients to pass into the body but prevents food particles and bacteria that could cause harm. Certain cells that line the intestine, including enterocytes which absorb water and nutrients, goblet cells which secrete a protective layer of mucus on the intestinal wall, and Paneth cells which secrete lysosomes that contain digestive enzymes, help to modulate this activity and keep a healthy balance. In their study, which published in the journal Laboratory Investigation, the researchers found that molecules in broccoli called aryl hydrocarbon receptor ligands bind to aryl hydrocarbon receptor AHR, which is a type of protein called a transcription factor. This binding, they found, initiates a variety of activities that affect the functions of intestinal cells. To conduct their study, the researchers fed an experimental group of mice a diet containing 15% broccoli, equivalent to about 3.5 cups per day for humans, and fed a control group of mice a typical lab diet that did not contain broccoli. They then analyzed the animals' tissues to determine the extent to which AHR was activated, as well as the quantities of various cell types and mucus concentrations, among other factors, the two groups. The team found that mice that were not fed broccoli lacked AHR activity, which resulted in altered intestinal barrier function, reduced transit time of food in the small intestine, decreased number of goblet cells and protective mucus, decreased Paneth cells and lysosome production, and decreased number of enterocyte cells. The gut health of the mice that were not fed broccoli was compromised in a variety of ways that are known to be associated with disease. The research suggests that broccoli and likely other foods can be used as natural sources of AHR ligands, and that diets rich in these ligands contribute to resilience of the small intestine.